The following content is provided under a Creative Commons license. Your support will help MIT OpenCourseware continue to offer high-quality educational resources for free. To make a donation or view additional materials from hundreds of MIT courses, visit MIT OpenCourseware at ocw.mit.edu.
Today what we're going to do is we're going to kind of talk about some of the big ideas and I have my on. Oh, all right. Details. All right. Today what we're going to do is talk about pattern formation in biology, some of the different mechanisms that are employed in development and other contexts in order to, for organisms to kind of figure out where to put things. All right. Now, before I get into that too much, I want to just make a couple administrative type announcements. So first, we have graded the exams, and we'll hand them back at the end of class today. If I try to forget, then please somebody remind me. What we're going to do is we're going to start by just kind of talking about diffusion a bit more. I think that there's a fair range of different maybe experiences thinking about diffusion and what it can do for you. So we'll work on our intuition a little bit. And then I'll say something about the reading that you did from Uri's book about kind of robust mechanisms for pattern formation in the context of development. All right, so simple diffusion with degradation leads to these exponential profiles that are not robust to changes in the concentration or the production rate of the morphogen. Whereas if you have self-enhanced degradation, then this leads to a power law kind of fall off that does have some that's more robust to the concentration of this morphogen. Then what we're going to do is transition to these Turing patterns, reaction-diffusion systems, where you can have a really surprising effect, whereby if you, in a well-mixed situation, these chemicals or proteins or reactants, they might just reach a stable state. Okay? However, once you add diffusion, then you can start getting pattern formation, which is a very funny thing because diffusion is normally something that smooths out profiles. So normally we think about diffusion as being something that removes patterns. And indeed, that's generally what happens. But in this, following from this classic work from Alan Turing at the end of his life, he showed that it's actually in principle possible to have the emergence of patterns from diffusion. And we'll talk about some of the ways that can possibly happen. Interestingly, all right, so Turing patterns are already kind of a surprising case where diffusion leads to patterns. There's another interesting phenomenon where if you add another source of noise, that again, you think noise, for example, demographic noise, the random creation, destruction of individuals or chemicals or proteins, normally we think this should also kind of be a force for removing patterns. But there's been a number of pieces of work over the last decade showing that in some cases, demographic noise can actually lead to patterns, either in space or in time. We're going to talk more about this in the context of these so-called noise-induced predator-prey oscillations. But since we're talking about patterns here, I just want to I will tell you something about how such noise can enhance the formation of patterns, in the context of the Turing mechanism. And then we'll end by talking some about recent work of how E. coli find the center of their cell when they want to divide. All right, so you can imagine that this is not an obvious thing to figure out how you might do it. We'll tell you. We'll talk about the so-called MIN system that E. coli use to find the center of their cells, so they know where to septate, where to cut off its big two cells. Okay. All right, so I just want to start by making sure that when we talk about diffusion, we're all talking about kind of the same thing. Now, hopefully, for those of you that have not been thinking about diffusion so much recently, you did read the notes that Alexander van Oudenarde put together for systems biology a few years ago. I think it's useful, but in addition to the math, as maybe many of you know, I'm a huge fan of sort of graphical representations of ideas. All right, so so I just want to make sure that we all kind of agree on some of the basic ideas of how, for example, we get flux in the context of diffusion. Okay. All right, so imagine that we have a one-dimensional system where there's some chemical with concentration C. And we can imagine, for example, it starts out looking like this. Okay, so this is a concentration profile as a function of position x, where we have maybe a box of length L. Okay. Concentration of some chemical is a function of position x in some space. Okay. Now I just want to make sure that we're all thinking about the same thing. All right, so it starts out some concentration C1. Over here might be some C2. All right, linearly here. All right, I just want to make sure we all agree on the magnitude of the flux at some different points. Okay? So in particular, we can think about A, B. This is capital C. All right. So where is the magnitude of the flux largest? A, B, C, D corresponds to them being the same. E don't know. All right. Are there any questions about the question? Yes?
On the y-axis, you're plotting concentration?
Yes, this y-axis is the concentration as a function of the position x.
Okay.
All right, I'll give you another 5 seconds to think about it. All right, do you need more time? All right, let's vote. Ready? 3, 2, 1. OK, great. So I'd say vast majority here are agreeing that it's going to be D. This is all, the flux is going to be the same everywhere. Okay. Now the flux, can somebody remind us? We'll say the flux J. And what is it going to be? All right, so there's a minus a D times the change in C with respect to x. And so this was derived in the notes. All right, so this is highlighting that what leads to a flux is the change in the concentration with respect to position. All right, so it doesn't matter that the concentration is higher here than here. You have the same flux, and the flux is in which direction? Left, right, up, down. 3, 2, 1. Left. All right, so the flux is here. Okay. All right. Minus sign. Like always, it's hard to remember from equations whether there are plus minus signs, but you should be able to just remember you can draw something like this and say, OK, this is a positive dc dx, so flux is going to be in the negative direction, right? Okay. Now, what will be the change in the concentration with respect to time at this point right now? Is it, all right, so this is change in the concentration at point B with respect to time. Is it greater than 0, equal to 0, less than 0, or can't determine? Can't determine. All right, does everybody understand the question that I'm trying to ask? The change in the concentration at this point, with respect to time, at this time that I've drawn. I'll give you 5 seconds to think of it. All right, ready? 3, 2, 1. OK. All right, so this, again, it's pretty good, but not everyone. All right, so it is going to be B. Can somebody explain why it's B here? Yes?
There's as much flux coming in as coming out on the other side.
That's right. So it's true that these that there's a net flux here coming to the left, but from the standpoint, if we want to know whether there's a change in the concentration at that point, we need to know about what's the net number of particles moving to the left versus the net particle number that are coming in from the right. Okay. Now, of course, on this left face, if I draw a little box here, are all the particles crossing this kind of position, are they all moving to the left? No. Right? So the idea is that all the particle motion is random. And indeed, there's only a slight excess of particles moving to the left as compared to the right, in the sense that if you look at the concentration here, the concentration is rather large. It's only a little bit larger to the right than to the left of this plane, which means that there's only a slight excess of particles diffusing to the left as compared to the right. But that leads to a net flux of particles crossing this plane to the left. Okay. All right. So where will the concentration be changing? At this point here, will the concentration be changing with respect to time? Ready? 3, 2, 1. No. All right. So does it change at all ever anywhere in this example? Right. Is this a steady state profile? Yes or no? Ready? 3, 2, 1. No. Okay. What should be the steady state profile? Flat. Okay. So how does that come about? If the concentration is not changing anything, yeah. Yeah, it's only changing at the edges initially. Now it's important to note that it's not that the concentration profile is going to it's not going to start looking like this. Right. So that will not actually be how it, eventually it's going to be flat, right? But it's going to kind of smooth out on the edges, and then it'll kind of. Do you guys understand what I'm trying to say? It's not that it's a line that just kind of goes like this, but instead we do end up getting curvature and stuff like that. Okay. All right. One more of these, and then we'll consider ourselves to be expert diffusionists. Okay. All right. So let's say the concentration as a function of position looks something like this. Okay. All right. What I want to know is where is dc dt maximal? Okay. We have some different points. We have A, B, C, D. Five seconds. All right, ready? 3, 2, 1. All right, so we finally got a lot of disagreement. I like it. OK, turn to your neighbor. You should be able to find somebody that disagrees with you. What is it that determines? All right, did you guys all agree? Okay. But you all agree on it? OK, let's go ahead and reconvene. And let me see. It may be that we're forming domains of some sort. All right, ready? Wait, he says no. All right. OK, one question, yeah.
Which is, is this instantaneous in some sense? Is
yeah.
it exactly at the instant?
All right, so I guess what I would say is that right, this is concentration profile at some time as a function of position and time. And if you want to know the concentration profile sometime delta t later, right? T plus delta t, right? Well, then it's going to be the concentration that we had before, and then a little bit, plus delta t times the derivative with respect to time. Right, so I get, so it's at this, right, so this is the concentration profile at this time, and I want to know how much is it going to change in the next delta t. Yeah. And I'm assuming, of course, that I'm not adding any particles or taking any particles away, right? So just due to diffusion. Okay. All right, let's see where we are. Ready? 3, 2, 1. OK. All right, so it's we got domain, definitely some domains. All right, so it's some, OK, well, it's going to end up being A, as I think a majority of the group is now saying. All right, so, and why is it A?
Because the second derivative.
Second derivative, right? So just what we said before is that if we wanted to know the change in concentration with respect to time, at this point that was 0 because there was the flux of particles to the left, or the flux particle was leaving that point, and the flux coming in were the same, right? And that's because of, and we can also think about the second derivative. All right, so this indeed just from Fick's, I guess that was first law, we get d, so this gives second derivative, c with respect to x squared, right? So that's a 2. Okay. All right, what this is saying is that to determine how rapidly the concentration is going to change at a particular point with respect to time, we need to know the curvature of the concentration with respect to position. Okay. So many people were saying C here. And actually, this is where the concentration will remain exactly constant over this next delta t. Right? And of course, if we want to know about what the concentration is going to do for a long time, then we have to do something more subtle, right? But if we want to know how the concentration is changing right now, then we just look at the curvature. Okay? And indeed, the curvature here is maximal. Right? So concentration, dc dt at this point, is it going to be greater or less than 0? Ready? 3, 2, 1. Less than 0. Right? Now, it's true that only the curvature at a particular point matters to know how the concentration is going to change over this next delta t. But if you want to know about what's going to happen over longer periods of time, then you have to worry about more of the global structure. Because what's true is that this local maximum may quickly go away. Right? So you don't have to wait very long before this thing gets smooth, and then dc dt will the magnitude will go to 0. Right? So in some sense, this big hump will stick around longer, right? But because of the curvature, this has the maximal of magnitude of dc dt. Okay. Right. And it makes sense that the concentration is going to go down because the net flux here is to the right and to the left. Right? So there's a net flux of particles leaving. Concentration is going to come down. Okay. Now, are there any questions about what we've said? I'd say that if you find this discussion confusing, I would strongly encourage you to get together with a friend and just draw some random lines, curves, and just make sure you understand where diffusion is going to pull things and so forth. Okay. All right. No questions about that? Okay. All right. Whoa. All right, so I just want to say a few things about the so-called French flag model in development. So this is perhaps the most famous. All right, does everybody know what the French flag looks like? What's that? All right, yeah, I know there are 3 stripes in some orientation, right? Now, but the idea here is just that a simple way to specify kind of a structure across one axis is to have a diffusing chemical, or generally a protein, and then based on the concentration of that protein or morphogen, you can just have the tissue kind of read out what it's supposed to be, or what it's supposed to do. Right. So the idea here is that you have some morphogen that starts maybe on one end of an embryo, and it diffuses. Right. So we end up with some curve that tells us about the morphogen concentration as a function of position. All right, this is as a function of position. All right, so this is along, say, the embryo. Okay. Now, the general challenge in the context of development is asking, we start with this embryo with many different cells. They're all genetically identical, and they all start out maybe without any positional information. They don't know where they are, but what that means is they don't know whether they need to develop into either a head or a tail or something in between. Okay. Now, what often happens is that you can have deposition, often by the mother. So you have maternal deposition of, say, RNA that leads to expression of some morphogen on one end, and then it diffuses. And based on the concentration, you can say what part of the body you should be developing into, right? So the idea is you just have these thresholds, right? That might be some M1, M2, right? Now all the cells over here know that they should maybe develop into a head. Over here, they're going to develop into the mid-body, and over down here it could be the lower body, for example. Okay, so just from the concentration that a cell feels at a particular location, so if you imagine you're a cell right here, you just say, okay, the concentration of the morphogen is between M1 and M2, and then from that you know what you're supposed to be. Okay. Now, if you have no degradation within the body, let's just imagine you say let's say that at one end, 0, we're told morphogen concentration is M0. And let's say that the other end, we know that the morphogen concentration is going to be 0. Let's say there's a lot of degradation at an end. All right, if there's no degradation in between, what should the profile look like, given these? All right, so no. All right, so just diffusion with these boundary conditions. All right, I'm going to draw some options, and hopefully you can close your eyes and imagine what it should be. All right, we know that it starts out at M0. Over at L, it's equal to 0. All right, is it closest to A, B, C, or D? Do you understand what I'm asking? Yes?
This is steady state.
This is the steady state concentration profile of the morphogen given there's no degradation in the interior. Yep. Ready? 3, 2, 1. All right. Yeah, so we're, at least a majority are agreeing it's going to be B, although there's a significant minority that's saying A. Right? And this is why I'm bringing this up, because we're so used to seeing exponential profiles, we kind of think that you should always be getting them, and I want to be clear about why it's an exponential profile. That is the thing that we start with, and for example, this model in Nuri's book. Right? In Nuri's book, you get exponential profiles as a result of first-order degradation. Okay. Whereas if you don't have any degradation in the interior, then we say, okay, DC, D. Well, do we want to use C or M now? Okay. We'll use M just because now we're thinking very particularly for a morphogen. All right, so dm, dt, all right. Okay. We want to know the steady state profile. Okay. We set this thing equal to 0. All right. Just integrate twice. All right. So we're going to get the morphogen profile should just be some ax plus b. Okay. My line was kind of crappy. I'm sorry. However, when we have first-order degradation, then we get these exponential profiles. Okay. Are there any questions about?
Is
Yeah, right. So this would be this require. Yeah, what does this require?
Morphogen is generated at x
Yeah.
equals 0 and
Degraded at
disappear at.
Yeah, right. So that's what I was saying is that when I said that at the end it was a 0, means that we have degradation at this point. Right, so anytime that the morphogen reaches the end of the embryo, say it gets degraded. Okay. Right. So it's not degradation inside, but it's only degradation here. Right. And this would correspond to a situation where you have constant flux throughout the embryo, constant rate of production of the morphogen, constant rate of degradation over here. Okay. Right. Right. But when you have this first order degradation, right? So if we're told that instead, well, maybe. Right. So if in addition to having diffusion, you also have this first order degradation, right? Now remember, m is a function of both x and t, right? Then this is the situation where we get exponential profiles. Okay. Now in Uri's book, he often discusses this, where the boundary condition is that you have some concentration m naught at one end, and then this is m. And then we get an exponential profile here. Okay. What was the characteristic length scale? And this is something that we should be able to figure out. All right. You should, of course, be able to solve the equations, but you should be able to figure it out from our favorite approach of dimensional analysis. What's the units of things here? Okay, so units of d is what? Length squared over time. All right, and units of alpha? Right, so this is a morphogen concentration over a time. This is a morphogen concentration. So this thing has to be 1 over time. Right, so the length scale is then going to have to go as the square root of d over alpha, right? Okay. And indeed, if you solve it, it's exactly equal to that. But this dimensional analysis just told us that it had to scale like that. Okay. All right. So this is telling us about this characteristic length over which this morphogen profile is going to fall off. All right. This is the characteristic length L. Okay. Incidentally, you'll see many cases of things that look like this. All right. So if there's first order rate that something is either degraded or is uptaken or whatnot, then together with diffusion, you'll get an exponential profile with the characteristic length scale that's given by this ratio. Right? And so you'll see this in many different contexts of, for example, nutrients kind of going into a biofilm. Right? If you have cells picking up nutrients, then it's roughly kind of it's not that it's being degraded, but it's being imported. Right? Then you'll have a similar process. All right, so this is a nice, fine situation, except for what was the problem that Uri pointed out? It's not robust, and it's not robust against what? Initial condition, yes, but I think we have to be a little bit more specific than that. Right, it's not robust to changes in M0. Incidentally, in most contexts, I mean, I think for mathematical simplicity, it's useful to just have a boundary condition where m naught is constant. But in general, is that what the mother is going to be fixing necessarily? What would the mother typically be fixing? What's that?
Production rate.
The production rate, right? How do we figure out what the production rate is in this situation?
It's the flux.
That's right. The flux where?
Right at.
That's right. Right. So the production rate is the flux right at, or just to the right of 0. Okay. Incidentally, where is the flux maximal here? Is it maximal at A, B, or C? Ready? 3, 2, 1. You can do it verbally. Fine. A, right? And that's because it's the profile is steepest here and then shallows out, right? So flux is decreasing here, and that's because some of the morphogen is being degraded, right? Right. So indeed, we can figure out what the production rate, right? This is equal to the flux, right? Which is equal to minus dc dx, right? That location, right? So the morphogen profile, m as a function of x, is some m naught e to the minus x over L, where L is given by this characteristic length scale, right? So if we ask what's dm dx, well, it's going to be, we get a minus m naught over L. Okay. Evaluated at x equal to 0, just makes that go away. So we end up with this. So you can figure out what the flux is, right? Yeah, so it's just an extra couple of terms. It's fine. But that would be what would typically be constant, because there might be some kind of RNA deposit there, or if it's actually transcription off of the genes at this, in the cells in this location, then it's based on whether there's 1 or 2 copies and so forth. Okay. All right. So this is all fine, except that this thing is not robust to changes in m naught or this production rate. Whereas in many cases, what we see experimentally is that if we, for example, have the production rate, then the profile that we see out here is somehow remarkably similar. Okay. Yes?
In which system? Because I mean, in Drosophila, profile.
Right. Yeah. Okay.
What are the kind of model systems?
Yeah. Okay. So I'd say that in many cases, it's not that it's directly this m squared business, but there are a number of cases in Drosophila that where they actually do see this, and in many cases it was via this active it was a self-enhanced degradation, but not that it was directly. It was via binding to a receptor. So this was seen in patchless. Together, I want to remember which were the models that. I guess it was okay. I'm worried I'm always going to get. So it's like patchless, and frizzled. Or are there any Drosophila people here that came up with these crazy names? I'm trying to. All right. Yeah. So I always get confused about which are the morphogens and which are the receptors in these situations. But I'd say that it is something that's observed in a number. I'd say a number of different contexts, both in at different stages of development, both in Drosophila and they see this in the wing as well as in the body. So at different stages, I think it is seen actually. Yeah. But I'm very much not a developmental biologist, so yeah. All right, so the proposal that Uri suggests as a way to kind of make this thing more robust is to somehow change this term, right? Now, the goal in all this was to have some degradation rate, and he writes it as f is a function of concentration m. In principle, this thing could be a function of the position x, but he doesn't want that. And why was it that he didn't want that? Yes. Right. So the whole goal is that we want to start with some situation where we kind of don't know where we are as a function of position x, and then from that get some spatial information, right? So we really want something that's not a function of x in there, right? Because then we've already solved the problem that we're trying to address, right? Right, and so one proposal is indeed just to have something that, and this is very much a phenomenological approach, which is to have this thing be kind of have self-enhanced degradation, right? And right, so the idea is that if we have the Morphogen profile, that was looks something like this, and I just want to highlight the in the context of there's multiple ways you can have something that looks like this, right? So if along the way to degradation, if the morphogen actually binds a receptor that then activates the degradation, and this is seen, for example, in Drosophila. This is hedgehog and patched. So R is right. So in this case, you have the morphogen that's diffusing outside the cells. It binds to the receptor, and then the receptor activates important degradation of the morphogen. Right? So it's not that the morphogen is directly leading to its own degradation, but it's via some network here. And there was another one that they talk about, which is that when the degradation is mediated by two things that look like this, and this is in context, and this is again Drosophila, but you could probably guess what this thing does. Okay, and so in context of wing development as well, right? Yeah, so it's not that this has to be. If you write down an actual model of one of these things, and you could play with it and see where you're going to get something that's going to look like an m² type term. But the nice thing about this phenomenological approach is it kind of gives you a sense of the kinds of effects that you should be looking for that will enhance the robustness of the pattern. Okay. Now, the key thing in this whole discussion to remember is that if you have a profile here, if you change, that it's somehow translationally invariant in the sense that since there's no information about where you are here, these patterns can kind of slide over, right? So if you change the boundary condition m0, that's just equivalent to sliding this over some distance, and you could figure out where everything has to go, right? All right, and that's true not just for the exponential profile, but for any of the profiles, right? And that's just because there's no information about what's going on once you're inside the embryo. Okay? And can somebody kind of give the intuitive explanation for why some sort of self-enhanced degradation like this might make the pattern more robust? Decrease faster when m is big. That's right. And so the idea is that if you change the m0 here, for example, you double it, then with a self-enhanced degradation, you kind of quickly come back to where you kind of were, right? So there's some sense that if the pattern kind of quickly comes down, then you can get it being more robust, right? And in particular, this profile for large x and in kind of the proper regime, this thing falls off as some a over x squared. Okay. And indeed, one of the problems in Uri's book is that if this is just degrading as m to some power n, then you can calculate what happens here. But this is some sort of power law fall off. Okay. So of course, it's falling off rapidly here, but not as rapidly as if it were an exponential. Right? And this is just like in the case of the power law distributions of networks that we were talking about before, right? That for the exponential fall off, then it's just very, very unlikely that you'll find some node with many edges. Similarly here, it's you quickly fall to 0 here as compared to a power law type distribution, right? So that means that you can specify patterns out further in some sense, right? Are there any questions about this basic idea and where it comes from? I think the discussion in Uri's book is actually reasonable. So I don't really want to spend too much time on it. In the context of the LDAR model, that was sort of the last section of that chapter, I think that it's one thing I just want to highlight is that it's useful to be able to look at these models, and from the equations that you see, for example, in the supplemental material of the paper, make sure you are very clear about what assumptions they've made in getting into this model, right? And so maybe we'll just spend a few minutes talking about this. All right, so this is, again, an Drosophila embryo, 2.5 hours after fertilization, where at that stage you really have the embryo that kind of looks like where we're trying to understand the patterning around the kind of the front back, right? So we think about diffusion along in one dimension, but it's really around this radial direction, right? And in particular, where this is trying to understand patterning in the dorsal region, okay? And what we have is a situation where we have this protease, the inhibitor. Right, so the protease is kind of distributed uniformly. The inhibitor starts out there, and then we have the morphogen that somehow starts out in here. And we want to end up with a situation where the morphogen profile in here is kind of well defined, right? So where you just have morphogen at the center of this dorsal region. And then we have diffusion of these inhibitors coming in, and then the morphogen coming out. And so the question is, how can we make sense of this, right? And if you look at the equations that they write down, right, so we have diffusion of the inhibitor. This is the complex of the inhibitor and the morphogen, okay? Complex C. Oops. Right, so this is a case where they wrote down the basic, what they basically knew about the biology of the system, and then they asked, well, what range of parameters would lead to a robust pattern of the morphogen in here if you varied things such as the total concentration of the inhibitor, the morphogen, or the protease. Okay. All right, so we have diffusion of each of these things, the inhibitor, the morphogen complex. We're not going to worry about the protease because this thing is uniformly distributed. All right, you can argue about whether that's the right thing to do, but that's at least in the model. All right, so we have diffusion of each of these guys. All right, can somebody say what's going on here? And what are these things trying to capture? Yeah, John. Right. So this is just saying, OK, the complex is formed as a result of binding of the inhibitor and the morphogen. It's proportional to the concentrations of those two things at that particular position at that particular time. Right? And just remember that any time that you create something, if you have binding of two things together, then you have to consider these things going away. And it's not that they're being degraded. It's just that they're forming this complex. Okay? So in these equations, you just have to be very careful about keeping track of which things are really going away and which things are just changing form. Yeah?
So the chaos is zero in this case, like when the inhibitor binds to the morphogen, like they never unbind.
Right. So this is, I want to be, you're saying why that there's not a minus term. Yeah, so this is an assumption. Right. And I think that this is the kind of thing that right? You have to be very clear about any of these because that's always going to occur at some rate, right? And what the assumption is that this is dominant, okay? Right? And just to be clear in words, what is this thing? Right, it's the protease, all right, and you're saying cleaving the inhibitor, and
yeah, cleaving the complex by degrading the inhibitor.
Right, so at a rate proportional to the protease concentration and the complex concentration, this complex goes away, right? Now the question is, we have to figure out where it kind of went or what happened, right? Well, and we have a minus alpha C here, but then we have a plus alpha C here, but this is at the morphogen, right? This is at the rate that the complex is being somehow is being disappeared by the protease. The morphogen is appearing, right? But then we don't have the same term here, right? And that's how you know that what the protease is doing is it's degrading the inhibitor as part of the complex, right? So by looking at these equations, you can figure out what are the assumptions that have been made and what the biology is that's being captured here. But in many cases, there are assumptions about what's big and what's small. Right? And then what's this thing here? Somebody else that has not yet? Do these equations look familiar? All right. Yes, please. Yeah, that's right. It's the protease degrading the inhibitor when the inhibitor is not bound to the morphogen. Okay. All right. Now, of course, what the authors did next is they kind of searched numerically across parameter space over forward orders of magnitude, and they found that for some parameter regime, there was a robust pattern of the morphogen developed in here. Right? And there's no reason for you to necessarily remember which thing it was, but what they found is that if this term kind of went away and this term went away, then you end up getting a robust profile of the morphogen against changes in the overall morphogen or inhibitor concentration. Right? Now this is pretty weird, I would say. Right? So I don't know. Do you think is this one weird? I don't know. I mean, weirdness is in the eye of the beholder. But I would say it's not so shocking. I mean, but this is saying, this is saying that well, can somebody say verbally what this would correspond to?
Protease doesn't degrade the inhibitor on its own.
That's right. The protease doesn't degrade the inhibitor on its own, only when it's part of the complex with the morphogen. Okay. And actually, apparently, there was experimental data already indicating that was true. So you could have, if you wanted to, just written the model without that, because there was already evidence for that. But I'd say this is a pretty weird. This is a pretty weird thing, right? Okay, and so this would be saying that for some reason, diffusion of the morphogen on its own is very small. Okay? And in particular, small compared to diffusion of the morphogen when it's complexed. Right. Now, can somebody say why I might think that's weird? I mean, you don't have to believe it's weird.
Make something bigger and it diffuses better.
That's right. Yeah, exactly. Right. So this is saying, well, when it's complex, it's bigger, it's somehow diffusing faster. Right. So this would not happen due to simple Stokes type drag. Right. This would have to be this would be biology. Right. Okay. No, but I mean, but in some ways, biology is allowed to do anything that doesn't violate the laws of physics, right? Indeed, later there was experimental evidence that something like this was actually happening, and you can imagine it happening either from sort of active transport type dynamics for the complex or from binding type dynamics of the morphogen. Right? But I think the key thing to take away from this example is just that. In general, we would like biological function. Well, biology would like biological function to be robust to things that are often fluctuating or varying, right? And that's one way to try to make guesses about what might be happening in the system. This sort of computational exercise is not at all a proof that this has to be what's happening, right? They wrote down a particular model. It could have been that there are other terms they're not aware of and so forth. But it's at least a way of generating hypotheses that you can go and test. Okay. And quite generally, I would also say that it's essential for all of us as consumers of models to be able to look at somebody's equations and figure out what it is that they've assumed. Okay. All right, so with that, I want to move on to this idea of pattern formation via these the reaction-diffusion or Turing type patterns. And I think it's really important to start by just acknowledging that this is really a surprising finding, that it's even possible for this to happen. Okay? Because this is a situation where you have a couple chemicals or proteins or reacting elements, and if you just mix them, if you have them in some well-mixed tube, they reach some steady state. Okay? Whereas somehow, if you allow diffusion, then you can get these patterns, right? And this is, and I would say, based on my intuition at least, I would not have thought that this would be possible. Okay? And this really just is because if you just imagine you start with some profile. Now there's some structure here, but diffusion is going to naturally cause this to come down and this to come up, and right, so diffusion acts to remove these spatial patterns. Right? But somehow, in some parameter regimes, if you have coupled reactants that are they're activating and inhibiting each other, then you can actually get these spatial patterns. Okay? And as you read about, there's some idea that you need to have kind of a local activation and kind of a global inhibition. But ultimately, the mathematical, you always have to go and think more carefully about the math than would be indicated just by those words. So this is at least a way to guide your thinking a little bit. But ultimately, what matters are that, around this stable, what would be the stable fixed point, you have to actually look at these derivatives and so forth. And the derivatives can be subtle, I would say. So it's not just because you think of something as an activator or inhibitor doesn't mean that it's going to have that role around the fixed point that you're studying. So this is just a caution, I suppose. All right. So what I want to do is just give you, for example, one example of a simple model that does experience these Turing patterns in an appropriate regime. And this is the Levin-Segel model of pattern formation. All right, and this was published in 1976, and it was actually meant as a model of kind of predator-prey interactions in ecology. We'll talk significantly more about predator-prey interactions in a few weeks, but I just want to write down what they said. And this is a simplified model of their stuff. So I was actually trying to think about some plankton herbivore interactions. All right, and this is derivative with respect to time. Okay, we're gonna follow the nomenclature of a paper by Butler and Goldenfeld a few years ago, because they're the ones that thought about the demographic noise enhancing the patterns. All right. All right, so we have phi and phi. Okay. First of all, who's eating whom? All right, we're gonna say it verbally. Who is the predator? Ready? 3, 2, 1. The herbivore. Well, all right. So herbivores are not what they used to be. Okay, right. So indeed, the herbivore benefits from the presence of the plankton. Right. So nobody cares about plankton, I guess. All right. So right. So this corresponds to the predator-prey type interaction. All right. So there's some death rate associated with the herbivore or the predator. And then what you see is that there are these two growth terms for the plankton. Right, so there's this term, which would be kind of simple exponential growth. And then there's this term, which is actually some sort of super exponential growth. Right? So there's some sense in which the plankton benefit each other. And this was originally introduced because of something called predator satiation. But it's just a general reflection of the fact that in many cases, individuals benefit from the presence of other individuals. Right. And in particular, this is known as the alley effect. And we'll spend time talking about this in a few weeks as well in the context of populations and ecology. But for now, I just want to use this as an example of a model that gives you these Turing patterns. Okay? All right, so if you want to, you can go ahead and ask, well, what happens if we just have a well-mixed situation? All right, so if everything it's not a function of x, but it still can be a function of time. All right, you can solve these equations, and you can find what the steady states are equal to. Okay, so there is indeed a steady state, stable coexistence of the predator and the prey in a well-mixed situation. Right. However, if you then go and you analyze the stability of different spatial modes, what you'll find is that in some situations, particular wavelengths or wave vectors become unstable. Right? And it's just over some range of wavelengths, and that corresponds to the wavelength of the Turing patterns that you'll see. If you're curious about these things in more depth, I encourage you to attend Mehran Kardar's class, Statistical Physics and Biology. He is an expert on these topics and I think is a wonderful, clear lecturer. All right, now, there's going to be some condition for these things leading to Turing patterns. Okay. Now, from your reading, do you think it's going to be mu nu? All right, so this is Turing patterns require. All right, I'll give you 30 seconds to think about what. All right, do you need more time? I'm not sure where we are. Right, maybe another 15 seconds, just to, All right, let's see where we are. Ready? 3, 2, 1. OK, we're all over the place. I think we're uniformly distributed between A, B, and C. OK, so there's an idea that you're supposed to have so-called local activation and global inhibition, right? All right, why don't we turn to a neighbor and see if you can figure out what's going on.
Yeah, that's exactly. That's what I'm trying to. So like.
All right, why don't we reconvene? I'm curious where your thinking is. All right, let's go ahead and vote. Ready? 3, 2, 1. OK, so we have many, many C's. The idea that so mu has to be much less than nu. Yes. All right, I want to make sure. I was busy doing other things. OK. Right, and all right, so mu is telling us about the diffusion coefficient of the prey. Nu is the diffusion coefficient of the predator, right? So what you want is to have local activation, right? And the thing that's activating itself is the plankton, right? So you can see that that's this, like, for example, phi squared term. And from the standpoint of a spatial situation, you can say, all right, well, let's say that we have some region with a lot of this prey. Right? Well, it's able to activate itself, so it kind of comes up. And as it does that, it is creating more of the predator, phi, right? Okay, but because the predator has a larger diffusion coefficient, it's obviously going to grow here, but it will also diffuse away a fair amount. And that's this global inhibition of neighboring regions. Now, of course, these are just words. You have to take it with a little bit of a grain of salt. All right. But if you can actually do this calculation analytically and derive in this model the condition for Turing patterns to emerge as a function of everything. And for example, if you have b1 half, b1. All right. So order unity type parameters. Then the requirement to get Turing patterns is that nu over mu is must be greater than 27.8. Okay. All right. The key thing, though, is that this thing is much larger than 1. Okay. All right, so a general thing that emerges in these Turing patterns is that you need this so-called inhibiting type partner to have a much larger effective diffusion coefficient than the activating partner. Okay. Now, the problem with this is that this thing is indeed much larger than 1, okay? Which limits the overall maybe usefulness of this as a mechanism, just because if it really is simple diffusion, right? How much bigger would let's say low Reynolds number regime, right? These are just molecules experience diffusion. How much bigger does the inhibitor have to be? No. How much bigger does the? Which one's? Oh, this is so confusing. All right. So this is big and this is little, right? OK. Yeah. So how much bigger does the activator? Oh my goodness. There are too many things to keep track of here. OK. It's because I haven't even written down. OK, so that's the activator. OK, all right, let's just be, OK, so this one's the activator, right? And this one's the inhibitor. All right. And the inhibitor has to diffuse much more than the activator. So the inhibitor has to be much smaller than the activator. OK, good. So how much bigger does the activator have to be than the inhibitor for this to be true? Does it scale as this 28 or 28 squared or 28 cubed? Like, in terms of radius, how much scales linearly? Remember our Einstein equation, which you guys had fun thinking about on the exam, kt over gamma. All right, yeah. So this would require, for example, the activator to be 30 times bigger in terms of radius than the inhibitor in order to have this emerge just as a result of diffusion, right? Simple diffusion. And that's just not a typical thing. Okay. for just range of protein sizes. Okay? But you can imagine cooking up various ways to make this happen. But there's a very nice development that I mentioned, which is that there's another surprising aspect in these problems, which is that in many cases, demographic noise can lead to patterns. Or maybe you might call them quasi-patterns. But things that, for all intents and purposes, look like something that's pseudo-periodic. All right. So it's a pseudo-Turing pattern, maybe. Okay. And indeed, if you actually do this simulation with those parameters, right, where you do the explicit demographic fluctuations, i.e., you take into account that this corresponds to a prey giving birth, this corresponds to a predator eating a prey, random events, just like what we've done in the context of a Gillespie simulation. Okay? Then what you find is that with demographic fluctuations, or demographic noise, then the condition here, nu over mu, has to be, it's only 2.48. And indeed, in Butler and Goldenfeld, they derive these things using kind of field theoretic approaches. There was a 2011 paper and also 2008 or 9. Okay, right. And the nice thing here is that what you see is that in the presence of this demographic noise that will be there, the difference in diffusivities that are required is not nearly as large as when you're thinking about the mean field equations. Okay. All right, so just in the last, are there any questions about this before we talk about center fighting in E. coli? Yeah.
So now adding demographic noise doesn't fix the problem that the plankton needs to be bigger than the herbivore?
Oh. OK, now you're thinking about the actual plankton herbivore. All right, so I think that there are many things to say. I mean, one of them would just be that maybe the patterns that you see between actual plankton and actual herbivores is not due to the Turing mechanism, right? Yeah, right. But you still, I mean, for the Turing mechanism to be at play to generate spatial patterns, it requires that that this inhibitor do something equivalent to diffusion more rapidly. It has to move away more rapidly, right? But it could, that's the thing. It could be that the plankton only move around from the currents, whereas the herbivore actually has a directed motion. I have to say I don't actually know enough about plankton to know whether this is at all. I mean, I think that base statement I think is true, but I don't know how the numbers work out. Yeah.
You have like a one-sentence explanation why having stochasticity?
Yes. Yes. And both in this case and in the simple predator-prey oscillations, right? So this one thing that we're going to see with the predator-prey populations later is that you can also get this sort of oscillations in time, right? So this could be in time, or this could be as a function of space, right? But in both cases, it's really that you have a resonant enhancement in some areas. So the demographic fluctuations somehow excite all frequencies or all wavelengths. But then what happens is that, in this case, if you look at the eigenvalues, OK, actually, I want to be close to 0, though. If you look at how the modes decay, so the eigenvalue is a function of the wave number, what you see is that you end up getting things that look kind of like this. So whereas a Turing pattern is when particular wavelengths or wave modes actually become unstable, right? And then you get mean field type patterns. And in this sort of situation, what happens is that you're just close here. So then you excite all of the wave numbers or wavelengths, and then some of them take a long time to go away. So then they build up, and then that's the resulting patterns that you see. All right, so what I want to do is just, for the last 10 minutes, talk about this MIN system, because I think it's a beautiful example of a combination of kind of systems biology questions together with reaction-diffusion systems, and also some beautiful in vitro experiments that that I've quite appreciated. All right, so so the question is, you imagine you're a cell. Okay, you've gone to all this work. All right, so now you're nice and long. So you'd like to divide into 2 cells, and the question is, how do you know where to divide? Okay. So you imagine that you're some E. coli cell. All right. So you're maybe 6 microns long. All right. Everything is great. And the question is, you want to divide? Where would you like to divide if you're an E. coli cell?
Middle.
Middle. OK, so what you'd like to do is go here. Indeed, what happens is that there's this so-called Z ring. All right. There's a kind of a pseudopolymeric protein that forms a ring around here, and then it constricts itself and pinches off the membrane, and that's how you get 2 cells. Okay. So there's this formation of this Z ring that constricts, and that's this cell division event. The question is, how do you know where to put it? Okay. And that's what this MIN system is for. And it's, of course, like everything in bacterial genetics, it was identified by mutations. So, so the MIN system is called that because these mutants formed so-called mini cells. Right, mini cells. Right. So if you, instead of dividing in the center, where you might have 2 copies of the genome here, if instead you divide over here, then what you're going to end up with is a very long cell with 2 copies of the genome, and you're going to end up with this mini cell without any DNA. Okay, and those mini cells, are they going to do well for the long term? No. Right. And they actually can actually survive for a little bit, and people have argued that maybe they could be useful for synthetic biology because they still make proteins, but they're not going to go and take over the world because they don't have any DNA. But these are the so-called mini cells that happen if you have mutations in the MIN system. Okay? Now, I think the 3 players are MIN C, D, and E. MIN C, MIN D, MIN E. All right, so I think that MIN A and MIN B ended up not actually existing or something. I mean, in the sense that they identified the mutants, but then they were wrong about something. Okay, so the 3 that actually ended up being involved in the actual MIN system are C, D, and E. Right, so this MIN C prevents formation of the Z ring. Right. Now MIN D and E are kind of the amazing guys. So this guy binds to the membrane. And recruits MIN C. Whereas MIN E, what it does is it pulls MIN D off of the membrane. Okay, so it binds to MIN D and ejects from the membrane. And what has been seen by doing imaging in live E. coli cells with fluorescently labeled MIN D and MIN E is that there are remarkable oscillations from pole to pole. Okay? So, and indeed, that doesn't require MIN C. So MIN C is somehow following the others. What happens is that MIN D binds over here, then MIN E binds and kind of pushes it off, and then the MIN D comes over here, and then MIN E kind of pulls it off, and it kind of goes back and forth. The period is sort of minutes, maybe. Okay. So it's a really remarkable thing that happens in vivo. And of course, and the idea of what's happening is that if MIN D comes here and then it comes here, right, on each of the edges, then it doesn't hang out in the middle. And that means that it's only in the middle where MIN C can then bind, or sorry. No, so because MIN C is following MIN D, then only in the middle can you form the Z ring, because that's where MIN C is not. Right? Now, it's wonderful to do things in live cells because that's the native context and so forth. But I think that in some cases it's also wonderful if you can take purified components and recapitulate interesting behaviors in vitro, right? Because then at least you know what is, what's sufficient to generate a particular kind of dynamical behavior. All right? And because this system had been proposed as a model system for reaction-diffusion mechanism to get this behavior. And so there was this paper by Martin Luce in Petra Schwill's lab in, well, where was it? Yeah, in Dresden. And what they did is they took a supported lipid bilayer, okay, so a membrane on glass, and then they added purified components of min D and min E that were fluorescently labeled, and then they saw amazing patterns, these amazing reaction-diffusion waves traveling along. Right? Now, on the first day of lecture, I actually showed you what some of those things looked like. I didn't want to use up the projector again, but maybe I can pull up this movie because it is kind of fabulous. All right, so it may be that you can't see these things very well, but this is min D, min E, and an overlay of the two, imaged on a two-dimensional membrane. Okay, and the movies are just amazing. When you see this, you think that it's like a simulation, right? It's so incredible watching these things go. After class, you can come up. I can show you the paper, and you can look at the movie in more depth. But they're really amazing patterns, and it's patterns that you would predict from some sort of Turing-type reaction-diffusion mechanism. Okay, so there are a few things that are maybe worth saying in this business. So these are fluorescently labeled min D, min E, right? All right. So they started out with min D that was uniform, and then they added min E, and over the time scale of about an hour, they started seeing these sorts of patterns. Okay. As may be kind of expected from the in vivo behavior, what you see are these things where where min D looks like this, whereas min E looks like that. Okay, and then the wave travels here to the left. Okay, because the min E is ejecting the min D from the membrane, causing this whole thing to move. Okay. All right, so this is a situation where you have both these proteins in the liquid, right, so in the buffer, as well as on the membrane, and they're coming on and off and so forth. And in this situation, it's wonderful because they can do all sorts of things like control the concentration of min E and see that the velocity of these waves changes as you change the concentration of min E. Right? So this is a really experimentally tractable system. And then you can ask, well, what kind of model would lead to that sort of behavior? Right? All right, so such waves, do you think that it requires ATP? Just for fun, we can vote yes or no. It's all right. All right, ready? 3, 2, 1. Yes. Yeah. So indeed, it does require ATP. Okay. Because these things are, and that's a general feature of these Turing-type patterns, is that there is that is a non-equilibrium structure formation. Okay. Now, one nice thing that you can do in this sort of system is you can ask, well, what happens if I photobleach a particular area? Okay, so I come in, I photobleach. So now the profile looks like, I locally deplete the fluorescence here. Right? Right. The question is, will this move together with the traveling wave, or does it stay fixed? Right. And in these mechanisms, do you think, should this locally depleted area, should it move or should it stay where it is? Do you understand the question? All right. All right. Move, yes, no. Ready? 3, 2, 1. It actually doesn't move. Right, so depleted area doesn't move. And this is just a reflection of that these waves are not the result of individual molecules moving together with the wave, but it's like a wave like what you see in the ocean or along a string or whatnot, right? So the motion is a result of the individual molecules coming and going and communicating with each other, but it's not a reflection of actual molecules having directed motion. Okay? Because indeed, there's no mechanism in these models for directed motion, right? What you see is that in all these models, the only thing that is changed, that is a function of position are these is diffusion terms, right? So it's all random diffusion, but then you get global motion.